Greetings, ladies and gentlemen. Today I'm going to talk to you about prokaryotic cells. To start off, I'm going to tell you what prokaryotic means and give you one example and explain to you the 4 key characteristics of prokaryotic cells and some of the key structures of prokaryotic cells. To end off this video, I will also explain how reproduction in prokaryotic cells happens or occurs. So, prokaryotic. What does prokaryotic mean? Prokaryotic means before the nucleus was formed. So, pro means before and karyotic means nucleus. So, before the nucleus was formed. So, hence, the diagram, it has no nucleus label. And this leads us to our first key characteristic, saying that genetic material in prokaryotic cells are not membrane bound. Our second key characteristic says that there is no membrane bound organelle. So as you can see, they're all actually open organelles in there, and they're actually very little of organelles. There are less of organelles in prokaryotic cells, which leads us to our third key characteristic, saying that prokaryotic cells are small and lack specialization. It's because of this decrease and less in numbers of organelles, they have no specialized functions. And due to this lack specialization, it leads us to our fourth key characteristic, saying that most are unicellular. So because of this lack of specialization of cells, they pretty much carry out all 7 life processes as a single unit of cell. So let's continue by labeling. Well, I've labeled it, but then explain what these labels of prokaryotic cells mean. So to start off, I'm going to start with cell wall. Cell wall is this white region between the inner cell and this striped outer cell. They're not actually striped, but for easy representation, this white thing is the white section of the cell wall. Well, it is the cell wall. It actually protects the cell and maintains its shape like in plant cells, but they do not contain the same composition as plant cells. In prokaryotic cells, they actually are composed of a carbohydrate protein complex, which is called a peptidoglycan. So peptide means from proteins like peptides, and glycan like glucose from carbohydrates. So, and our next one, which is the plasma membrane. Plasma membrane is just this thin line here, as you can see, that like every other plasma membrane, it controls what goes in and out of the cell, and it's just inside the cell wall. So the cell wall is actually this layer, and the plasma membrane is just this line here. Our third key structure is the capsule. Not all prokaryotic cells have capsules. Capsule is just an additional optional layer which is made up of polysaccharides, and it actually helps the prokaryotes to adhere to structures such as teeth and skin and food. So most prokaryotic cells are actually bacteria, so when you get bacteria, when you're infected by bacteria in your mouth, they actually stick to your teeth because of these capsules. And our fourth structure is the pili. As you can see, these hair-like structures here. They're actually called pili. They're just hair-like projections, but they're little ones, and they actually help in the sexual reproduction of prokaryotic cells. So they act like a tube. They connect one bacterial cells to the other, and they transport this DNA from one prokaryotic cell to another, hence sexual reproduction. Our next hair-like growth, which is a longer structure, is called flagella or flagellum. This only helps in the mobility of prokaryotic cells. So like in sperm cells, it acts like a tail of the prokaryotic cell. Our next key structure is the nuclear DNA, so the genetic material. As I've said before, genetic material is not membrane bound, and prokaryotic means before the nucleus was formed. So prokaryotic cells have no nucleus. So this genetic material, which is here, this region that it's in is called the nucleoid region. Hence the name of the DNA called nucleoid DNA. This nucleoid DNA is the main DNA that helps in the phenotypes of help how the cell looks and how the cell adapts to environment. So its survival codes is in the its main genetic code is in the nuclear DNA. Usually this is the DNA that actually gets transferred during sexual reproduction. And the other type of DNA, as I said, is the plasmid DNA. It's this small circular structure. Well, not only plasmids are actually circular, they just look circular, but even the nuclear DNA are circular. So it's just, they're very, very long, so they have to be coiled around, but they're one single loop of DNA. The same as plasmid, they're one single loop of DNA, but they're actually really, really small. And these plasmid cells are actually of no use to prokaryotic cells. It may help them under unusual circumstances, but it's actually most useful to us human beings because we've used this plasmid for genetic engineering, like for the production of insulin and glycogen. So I've explained all the key structures of this. So there's 3 layers to the cell. So there's the plasma membrane, that's a thin membrane that controls what goes in and out of the cell. There's the cell wall that protects and maintains the shape of the cell wall, and it's made up of a carbohydrate protein complex, unlike the plant cell. And there's the capsule, which is an optional additional layer. Not every bacterium has it, and it's made up of a polysaccharide and helps in the stickiness of the bacteria. And the hair-like growths are villi and flagella, where the villi actually helps in the transfer of DNA during sexual reproduction, and the flagella helps in the mobility. And the 2 types of DNA are nuclear DNA and plasmid DNA, where nuclear DNA is present in the nuclear region and helps and is the main DNA that controls all the functions of the cell, whereas the plasmid DNA is not really of use, and it may help under unusual circumstances. And the one last important organelle or key structure of the prokaryotic cell is ribosome. Ribosome is the site of protein. So that's where protein and polypeptides are made, and it's where mRNA is translated into polypeptides. So that's the only thing different. That's the last organelle, and it's different to animal cells and plant cells. They have a different kind of ribosome. They're actually free, and they're called 70S ribosomes. So let's move on to how prokaryotic cells divide. So this process of division or reproduction of the prokaryotic cell is called binary fission, which is an asexual method of reproduction. And because it's asexual, and well, most prokaryotic cells reproduce asexually, such as E. coli, and it's because they're reproducing asexually, its daughter cell is genetically identical, and so there's not much variation in prokaryotic cell. Now, what happens in the binary fission is that the DNA, the nuclear DNA, is copied, and it divides, and it goes to the opposite poles of the cell. They usually stick to different areas of the plasma membrane, and because of this, they pinch off. So binary fission is actually pinching off. So this nuclear DNA, they actually replicate, they make a copy of themselves, and they move to either side of the cell. And so this cell elongates and become bigger until um, the signal for breaking down of pinching in the middle or cytokinesis is received, and all they do is they just pinch off and two cells divide. So they just cut off in the middle. And because it's just like every other cell dividing into two, they're genetically identical. They're using the same set of DNA. Now, well, if you see in the description box, there'll be two links, one showing a musical visualization of binary fission and the other explaining how E. coli actually reproduces asexually. So I hope you enjoyed my video, and please look at those videos link, and thank you for watching.